In this video, we're going to talk about the sense of olfaction, also known as smell. We've got these two outcomes for the video. So just like with all the special senses, in order for this, for us to smell something, we need to activate receptors, chemoreceptors. And that's going to be common if you haven't realized this already, depending on the order that we go through these. So chemoreceptors are activated by specific chemicals. Okay, we've got these chemoreceptors in our nasal cavity, and they're composed of bipolar neurons. Okay, this is a G protein pathway, and so just a quick review. Hormone, or in this case, a chemical binds to a receptor, so a ligand. Okay, GDP leaves, GTP binds, GTP and alpha leave and bind to a protein or an enzyme, or a protein which can be an enzyme or something else, and then downstream effects occur. Okay, so in this case, it's the similar pathway. It's the G-alpha S pathway. G protein receptor is activated in the cilia, so kind of those hair cells that are, they're hair cells, not the ones that you're thinking of, but you got hair cells in your nose or cilia. Okay, GTP and alpha are ultimately activated. They bind to adenylate cyclase, same as what we've learned before. This ultimately leads adenylate cyclase to produce CAMP from ATP. So remember, adenylate cyclase is the hammer, breaks down ATP into CAMP. CAMP is the key that we've now got that opens ligand-gated sodium channels. Okay, and as a result, sodium comes into the cell, and that cell becomes depolarized. And this is a graded potential. And wouldn't you know it, glutamate's released, right? When we talk about special senses, we're talking about, almost always, we're talking about the release of glutamate. Okay, this is the G-alpha S pathway. Now, what's different here is that we're not getting into protein kinase A. All we're saying is CAMP opens these sodium channels, that depolarizes the cell, glutamate's released. This leads us to something called combinatorial coding. Okay, there are 11 billion different types of things that we can smell. Okay, and taste, we can kind of generalize into 5 primary tastes, but even that, there are many, many different types of tastes. With smell, it's a similar idea. But what happens is with smell is that we activate a specific combination of receptors. Your brain gets this information, goes, oh, I'm smelling chocolate, because chocolate's on the brain right now. Okay, and so this happens because, we'll just say, chemicals from chocolate can activate multiple receptors inside your nasal cavity. Okay, these receptors are activated. Maybe some are activated a whole lot, some are activated a little bit, some aren't activated at all. Okay, and so then what happens is combinatorial coding takes all of this activation, inactivation, partial activation, and goes, oh, this is what I'm smelling. Okay, and so that pattern that's activated by that specific scent, your brain knows this is what I'm smelling. Last thing I want to do is just kind of go through the main pathway. Okay, so you have the olfactory epithelium. This is ultimately where those graded potentials lead to action potentials. Okay, then you have the cribriform plate. This is where these neurons go through in your skull. This leads to an olfactory bulb. This is kind of a relay. And so this connects your cribriform plate, or these neurons, and activate what we call mitral or tufted cells. You have the olfactory tract, which is essentially the highway that gets it up to your olfactory cortex and your limbic areas. This is where that processing occurs. Okay, the big thing, if I were to ask you about this on the exam, is just kind of big picture. So the cribriform plate is kind of the conduit or the connector. Okay, then you've got a relay, a highway, and then a processing center. Okay, but the big thing I'll focus on would be the combinatorial coding and just the G protein pathway. So there you go. What kind of receptors used for smelling, and what's going on with that G protein pathway?